Originally, people used to think that addiction happened because of withdrawal and that they were really part of the same process. And it turns out that addiction and withdrawal are completely independent processes in the brain. They occur in different areas of the brain, and as we've learned more about the effects of drug use, we've really come to a very different conclusion about addiction and withdrawal. So let's take a look at the research on how they seem to occur in different parts of the brain. So one area of the brain that comes up often with drug use is the ventral tegmental area, and that's because this is a major source of dopamine in the brain. It's one of the two major areas that produce dopamine in the brain. So, and we can abbreviate the ventral tegmental area as VTA. And it turns out that this area is one that rats will press a bar in order to self-inject morphine into that part of the brain. So we see that this is an area that seems to be associated with the rewarding aspects of a drug like morphine and the pleasurable aspects of it. And so the ventral tegmental area is associated with that aspect of drug use, the rewarding aspect, the part of it that sort of drives that motivation. And that's not terribly surprising because we associate drug use with dopamine activity, and so that makes sense. However, if you block opiate receptors in the VTA, you don't produce withdrawal symptoms. So enhancing activity here in the VTA seems to be associated with reward, but blocking activity there is not associated with withdrawal. So it seems to be an area involved in reward, but not withdrawal. In contrast, this other area, the PAG, periaqueductal gray, that we see here in the midbrain, is an area that's associated with withdrawal symptoms, but not the reward. In contrast to the VTA, rats will not press a lever to inject morphine into the PAG, and so it does not seem to have any rewarding or pleasurable aspect to activity in that part of the brain. However, once rats are addicted, blocking the opiate receptors here in the PAG produces signs of withdrawal. So we see evidence that the VTA and the PAG are really independent in terms of their roles in reward, in terms of the VTA, or withdrawal in terms of the PAG. So let's turn our attention to the reward pathway and learn a little bit about how this pathway is involved in influencing motivation for drug use. So the VTA is shown here, and it is a producer of dopamine. So the dopamine gets made here. The axons deliver that dopamine through this medial forebrain bundle to this area here called the nucleus accumbens in the forebrain. So it gets delivered from the midbrain to the forebrain, in particular to the nucleus accumbens here. So the nucleus accumbens is often an area that's associated with reward because the addition of dopamine to the nucleus accumbens seems to be involved in reward heavily. And then the nucleus accumbens, in turn, will spread activation through areas of the prefrontal cortex and frontal cortex. And that brings in another area that is key to the rewarding aspects of drug use and sort of increasing that motivation. So it starts to focus a person's motivation and goals and so on towards drug use. And don't think of this as something that only drugs do. This is how we're wired for a very important reason. If we did not have this ability to have our life experiences change our motivations and influence our motivations like this, we wouldn't have survived, because what this is a system where if we find food in a particular place, our brain kind of kicks off activity in this reward system, and it spreads this activation through the frontal cortex, essentially telling us, wow, that was really good food and I really enjoyed this, and now I'm motivated to go back here and find this food again. And if that works that way, that's good, because we're able to learn, we're able to do things that are rewarding. And if we think of what's rewarding, well, food, right? Water when we're thirsty, social companionship, all these things, sex, all these things are very rewarding. And so we're sort of hardwired to want to do these again if they are, rewarding like that. And drugs essentially just sort of tap into this system that is active all the time and that we use all the time to help keep us sort of focused and in touch with what we need to do at any given time. So it's definitely just sort of tapping into this reward circuit that is critical for human survival. I want to point out that there are actually two major reward systems in the brain. We can see here in this picture there is a system here in an area called the substantia nigra. And that's separate from this other system that we've been talking about that starts here in the ventral tegmental area and then goes to the nucleus accumbens and then spreads activation here through the prefrontal and frontal cortex, right? So that's the reward pathway that we see here in red, but then in green we see the substantia nigra pathway. And this pathway goes first deep into the brain into a set of structures called the basal ganglia, and that area is involved in regulating our movement. And so the substantia nigra dopamine system is implicated in movement as well as in Parkinson's disease. In fact, the substantia nigra degenerates, if you remember, in Parkinson's disease. And then the VTA, ventral tegmental area, is implicated in reward. And so dopamine is tied into both reward and movement. And again, that makes sense because we want to be able to move towards, locomote towards the things that we want, the things that we're motivated to gain. And dopamine projects to many areas in the forebrain, including the prefrontal cortex and the amygdala, and of course the nucleus accumbens, right? Associated with the feelings of euphoria, the pleasurable aspects of drug use, and other things. Reward, as powerful as it is an incomplete explanation of addiction. Over time, the amount of dopamine released to a rewarding stimulus seems to cease, and that reflects the effects of tolerance. We stop getting that big reward, right? If I have a slice of cheesecake every single day, well, by the 10th day, it's not that it doesn't taste as good, it's just that it's no longer quite as wow and rewarding. And more so, drugs really often stop being rewarding, and yet drug use continues. So there's much more to drug use than just people seeking a reward. Modern theory suggests that drug use is something that has been reinforced, and it reinforces drug seeking or other activities that are going on. So there's more of a reinforcement model that it sort of reinforces that behavior. Drug use can produce changes in the brain, and it's essentially your brain learning to make these changes in response to drug use. And so some of the things that we see are that there is an increase in the length of dendrites and the complexity of the dendrites. So sort of the types of changes that we would expect to see in a neuron that is being used, that is experiencing a lot of stimulation. And so we see these changes in the dendrites in the nucleus accumbens. And that seems to be one thing that's associated with drug use, is these changes in the dendrites, especially in the nucleus accumbens. There are also examples of there being hyperactivity in areas of the brain that are involved in learning and emotion when a person is experiencing craving. And so this might be triggered in a study by presenting a drug user with drug paraphernalia. So, some of the stimuli that are associated with drug use, it might be a pipe or whatever it might be. And so you present them with that, let them see it, maybe even handle it, and you can measure brain activity, and you do see that there's enhanced activity in parts of the brain when that happens, and that seems to be associated with sort of the craving. And additional brain changes are possible that are more pathological in nature. So actually malformed dendrites can be found, especially in the frontal lobes in some cases. There are some drugs, some medications that are approved or in various stages of testing for the treatment of addiction, and some are, more effective than others. For alcohol, there is this drug called Antabuse that's been around for a while. It's kind of a crude type of approach. It prevents digestion of alcohol, and so it makes the alcohol much more unpleasant and have more toxic side effects, including nausea. And so it sort of makes alcohol use, instead of a reinforcing or rewarding experience, sort of a negative and punishing one. There are some drugs that are thought to reduce the craving through actions on the glutamate receptor for alcohol. I don't know how widely used these are. They're not, how should I put it? They are not like magic bullets that seem to, fix the problem. For nicotine use, the approach has been to essentially do what's called drug substitution. So you substitute the typical nicotine source, for example, if it's a cigarette, which is really dangerous and harmful, for a form of nicotine that doesn't have the smoking aspect of it. So it could be a nicotine gum or a nicotine patch, and then you gradually reduce the dose and kind of wean people off of nicotine that way. There's a bupropion, which blocks the reuptake and reduces the nicotine the effects of nicotine on reward and withdrawal. So that has also been helpful. For heroin and opiates, there is Naltrexone, which is an opiate antagonist. It's going to block opiate receptors. So that is sometimes used in people after they've gone through recovery to prevent a relapse from having much of an effect. So by blocking the receptors, the opiate receptors with naltrexone, the person wouldn't really feel much of an effect if they took an opiate. Another approach that's done before a person is able to be completely free of an opiate is methadone, and that's an opiate agonist. So again, we're substituting a less harmful version of the drug for the drug itself. And so methadone is sort of considered a maintenance program. In other words, it substitutes for the illegal drug, and the person is able to kind of stay on that, and in doing so not be as susceptible to using, say, something much more dangerous like heroin. Buprenorphine is also an opiate agonist and is a little newer than methadone, and also works the same way, sort of as a drug substitution. And here you can see Suboxone is a drug you may have heard of. So that's a combination of buprenorphine, so the agonist that is going to keep the person from going into withdrawal, essentially, sort of substituting a legal and safer drug for something like heroin so that they don't go into withdrawal. And in addition, it contains naloxone so that they basically couldn't abuse the buprenorphine or find heroin, because the naloxone would prevent the effects. So it's sort of a combination dose where there's an agonist to prevent the person from going into withdrawal, but also an antagonist to prevent any abuse potential. Behavioral management for heroin addiction has a pretty low success rate, 10 to 30%. So, just typical therapy, type of programs have a pretty low success rate. But when combined with the methadone maintenance program where you're preventing the person or limiting the person's withdrawal symptoms, success rises dramatically to 60 to 80%. So really a much more successful treatment with these types of approaches versus the more traditional types of therapy. Drug treatment is cost effective. Addiction costs a lot, but every dollar invested in treatment saves about $7. So it's definitely, beneficial to continue to find approaches, including medications and safer drug substitutions for people who are dealing with addiction. And drug addiction tends to be lifelong, so these are not going to be quick fixes typically.